Okay, so this is what we are going to do today. We will spend some time to revisit the PCA using this teapot example. Then we are going to try the example we provided in the PowerPoint slides on Tuesday. This perception using dot matrix to do the operation inside the new perceptron and then to do the classification. I'm not so sure you have this deep neural network framework installed. So we are going to check, see whether your MATLAB support this toolbox. I don't have this toolbox in this computer here. So let's try what we can do with this. Then I will show you this classification learner, because last Tuesday we learned the classifications. We will play with small examples and to see how this classification learner application with the interface can help us to solve most of classification problems utilizing many different algorithms. Now I will show you in the toolbox how we can merge two datasets in order to be ready to compare. And I sort of still showed you how to do this customized marker based cell type annotation. If we still have time, I will see whether we can set up this environment to run Python and MATLAB together, because some of this function rely on the Python, external Python functions. So we are going to call those Python functions within the MATLAB to get the results. Okay, so let's start with the teapot example. So if you have this channel, you should be able to just copy this code, and we are going to set up. So if you copy this, the first row, for example, I'm going to do it now. This is a function which, let's see which teapot. So this is MATLAB provide example data, okay, for demonstration. So if you call this function, it give you 3D data points with X, Y, Z position, 3 of them. So what you can do if you do the scatter plot to plot each of these points. Say first column, second, just copy this, second column, third column. Okay, if you do this, you will get this teapot. Because each point has been predefined in that function. So it's just okay, to use this function, you can see the size, 10, smaller size, color, if you want to red. So you can have this customized variable to change the color, for example, smaller markers, or there's no categories, so every point are in the same group. So if you look at the center of this cloud, the state points, it's not centralized. If you found, for example, this position, it's kind of in the center. So its coordinates is negative 1.0.5. So this whole data point, this cloud of data point has not been centralized. What we can do is to get the centralized data and without change any shape. That's what we can do. We can do this row here. Oh, by the way, I add some noise, random noise to make this teapot example not so realistic. Just make things add some random noise around this point, so the points move around a little bit. So if you plot now, you can see the shape, but not exactly the perfect position because we add some noise. So our PCA will play with this noise, the teapot. It still has the same shape, but it's more like you random sampled some signals. Okay. Any questions so far? So this data is not centralized. It's not. We expect in the center of this will be average will be 0, 0, 0. However, this is a move to at least for the z-axis is 1.5 something. So in order to centralize this, we need to do this. Okay, minus is mean. Okay, so you have three columns. Define your x, y, z. They are not centralized, but if you this is uncentralized, but if you minus every coordinates average, then you basically move this cloud of data to the 0 center. So the outcome of V, so V will look like very similar to your raw data. So VERT versus this original variable, it's uncentralized, but now you have a centralized version. So if you plot centralized version of this using V instead of the original verts, you should get the same shape, and because the relationship between points has not changed, only you move the center to the 0, 0, 0. If you have if you can sample one of this position in the center, it's supposed to be centered around here. Somewhere, okay? Somewhere in here. And you see this is 0, and this 0, and here. Okay, so this is a centralized version. So we have a V here. So So PCA will play with this centralized version. So we run PCA on this V in order to get a rotation matrix. Okay, that's your previous lecture. So this output of the PCA, this will be rotation matrix. You have a 3D, so the rotation matrix will be 3 by 3. So this U will be a 3 by 3 matrix to help you to rotate your data. Okay. We also get this score, which is a rotated result from this built-in function, but we don't really need it. We can just compute ourselves. So in order to get this to rotate, you need to have this data and rotate with this rotation matrix in order to get the results. So that's what we are going to do, which is this one here. Okay. So S1 will be the rotated version of your teapot original. Now if you look at the, let's see, this original, let's figure, we put this hold on, because I'm going to draw the rotated version of the scatter. So what's the rotated version? This S1 contains a rotated version. S3. So because I have this old hold on, so I'm going to draw, I already draw this original before rotation points. By the way, it's centralized in there. And then I'm going to superimpose this rotated version onto the, and give it using different colors. Yes, so it should be S1. S1 is rotated version. So you can realize the PCA, it's not a dimensionality reduction technique. It's just found a rotation. So you still have 3D. What's the difference is this rotated version, this orange dots, will help you to display if you decide to use 2D to show them. So if I do this scatter instead of 3D, so that's a dimensionality reduction. I use 2D to visualize the first 2 dimensions of the rotated results. I hope through this example and through your homework, you hopefully understand what PCA is doing. Okay? So this is a rotated version. The PCA is the best to display the variability among your data points using 2D, although you have 3D information. If you don't do the rotation, you only look at original data using 2D. Original data is 3D, but you can say, okay, I only want to visualize a 2D. So that's your result. Okay? Before the rotation. This is a PCA solution 2D. This is before and after rotation. Okay. Yeah, what I found is this teapot has been rotated towards you. If you look at this blue points, they are teapots. Then the handle is here, and that's the top, and then since this PCA rotate to along this axis of x-axis. Okay, now I want you to try perceptron. So I want you to try whether you have this function installed on your machine. Do you have it? No, nobody has. Yeah, so this is a function neural network we have not installed on our system. So this is in a toolbox, which we haven't installed. But if you do have, you will be able to run this function, something like this. You have input data, and then you have a target to classify your input into 0, 1s, this classification. And you can train this perceptron and to get the trained model, and then you can predict if you have new data points. Okay, I think we are going to try the example we provide in this PowerPoint. We try this, okay? Let's put into the first data set. So we have 3 data sets. This big V here. So I'm just going to copy this and see I can put into it. So the teapot example is done. Okay. Let's call this input. So that's my input data. I have a x1, x2, x3, okay? In the PowerPoint, I have a V as an input. So this is a 3 training data. And I know the answer. My target is which I defined in here as -1, -1, 1, okay? So that's our target. So we hope our machine can take x in and give us -1, because I want to classify this as negative, this as negative, this as 1. So this is a 4D problem, because you have 4-dimension information to define each input. 3 inputs, okay, 3 target. So the idea is to have this perceptron in the middle. Then your x in and the prediction will be a t. And int x is 3 by 4. 4 by 3. 3 points, okay? 3 inputs. Then we want to have this vector to be target, -1, -1, 1. So what we need is a W, it's parameter combinations. W. So W will be let's see what 4 by 1. How to do this? 1 by 4. Okay, put this 1 by 4. So we can multiplicate W with X to get 1 by 3 output. We can make this 1 by 3. So we need to define 4 numbers here to set up this perceptron, to let it do the multiplications and give me the result I want. Okay. So I need 4 numbers for W. So I can see, okay, my W is a 4 random number. One by 4. So in here we have all 3. Let's see. Yeah. So the W, we have to have 4 numbers. Okay, so this is just random set. I just random set this perception using 4 numbers. My input is X. So what's the size of input? 4 by 3. So I need to use W times X. Okay. Yeah. W times X, because I know my input is 3 samples, each defined by 4 variables. So I set up my perception with 4 numbers to set this. So multiplication gave me 1 by 3. So this is the result. And because this is sign, so I use a function sign to hold this result. So my input is small x, okay? So for this example on the screen, the result is -1, -2, -1. So it's not a good result because my target, my training, I know it should be -1, 1. So what's wrong? It's because these values are not correct, not give me good prediction. So what we can do is just retry, generate another set of the random numbers, and do that again. Still not yet. Looks like this random number are not good options because they don't have enough range to give me the good results. So I need some negative values. So I'm using another set of random number. So this time using normal distributed. Try that again, and the result is opposite. So for these combinations, the result is positive, positive 1, negative 1. So still not good. So I have to try, keep trying. I try another random number set. This will give me 1, 1, 1, still not as good. Try another, still not as good. Okay, so this is a good combination. So I just try and different values here, and by chance, I get this particular set of the weighting schema or parameter combinations. Gave me the result what I need. So I can fix the values now. My values, it's trained already by these 3 inputs. So my training is done. So this W is fixed. Okay, I gave it fixed. That's my model. So next time, if I have a new data, say X input, X new, so I need 4 numbers. So next time I have this new data points comes in, I can apply my fixed, multiply it by this new. So my new input has been given the classification ID, which is +1 class. Okay. Because we don't have a neural network toolbox installed on all these machines, so I'm going to use my laptop examples to installation to show that. Before that, we can jump to classification learner. So if you do this, which classification learner, and let me know whether you have this installed. You should have already in the, because this is not a neural network toolbox application. It's within the statistic toolbox. It's not. It's by itself. The one I shared on the LinkedIn is example video. Yeah. So what we are doing here is we jump from the one which I just showed you. I'm going to show you again using the neural network toolbox. We jump to item 3. We are going to use classification learner to run a small example, so you see you can learn how to use it. We can let me jump to this video. Let me jump to this video to give you a very quick overview of this learner. Okay. So this is a very short introduction of the
models. Whether you're an expert or just getting started with machine learning, you often explore different types of models and repeatedly tune and test those models. The Classification Learner app in the Statistics and Machine Learning Toolbox lets you train classification models using supervised machine learning without writing any code. Classification Learner lets you perform common machine learning tasks, such as importing preprocessed data, exploring your data, quickly training multiple models, assessing model performance, and the iterative steps required to tune model performance, including optimizing hyperparameters, misclassification cost, and feature selection. You can find the Classification Learner in the Apps Gallery, or by typing Classification Learner on the MATLAB command line. Classification Learner lets you import data from matrices or tables. The app can automatically identify your predictors and response variables based on your data type. The next step is to choose the validation scheme used to measure model accuracy. Once the app has loaded the data, you can choose from several classification algorithms, including decision trees, logistic regression, support vector machines, and boosted trees. If you're not sure which to choose, the pop-up tooltip gives you a brief description of each classifier. Pairwise scatter plots let you explore your data for important predictors, outliers, and visual patterns or trends. Training a new model is easy. First, in the Model Gallery, choose one of the classifier presets, or the Train All option. Next, click on Train. The Current Model pane displays useful information about your model, such as the classifier type, presets, selected features, and the status of the model. For each trained model, you can see its accuracy on the History panel and inspect its misclassifications in the scatter plot. Look at the confusion matrix, and look at ROC curves available in the plot section of the tool strip. The confusion matrix lets you assess how the currently selected classifier performed in each class. Good classifiers have a dominantly diagonal confusion matrix, since all the predictor labels match the actual labels. Off-diagonal numbers indicate confusions between classes. You can force the classifier to reduce specific misclassifications by placing a higher cost on them during training. To do so, select Misclassification Cost from the tool strip. Specify uneven misclassification cost, and then retrain the model to see whether it helped reduce the undesired misclassifications. Based on your assessment of how a model may be improved further, you can use the advanced options to change classifier settings, or remove features with low predictive power, either manually or applying one of the many automated feature selection methods that are available at the command line. Instead of manually tuning model parameters, you can choose one of the optimizable models, and the app will try different combinations of hyperparameter values by using an optimization scheme that seeks to minimize the classification error or cost. The visualization shows how the classification error decreases as different combinations of hyperparameters are evaluated. Outside the app, you can also let MATLAB find the best optimized model in a single step using Fit-C Auto. After you create models interactively in Classification Learner, you can export your best performing model. Click Export. The model should appear in your MATLAB workspace, and its predict method allows you to make predictions on new data. You can also generate MATLAB code to train your best model on new data, or integrate trained models into computer vision, signal processing, or data analytics applications. To deploy on embedded devices, automatically generate C code for predictions, or generate a software component from your model for integration with standalone or enterprise applications. When working with Classification Learner, help is always just a click away. Simply navigate to the documentation using the Help button on the top right to find all the information you need. For example, the table here shows you guidance on choosing the right classifier, depending on accuracy, speed, and memory constraints. That concludes a brief introduction to Classification Learner. For more information, click on the links below. Thanks for watching.
Okay, so this is a very powerful visualization tool to help you to build the classification applications. You can build a function out of it. So our function, for example, for the perceptron here is just this fixed parameter combinations. And you can consider this becomes a predictor, okay? Of course, it's very simple. Then you have your input, then you can multiply your input with this parameter and get the results. But this is a foundation for the neural network and for your language model, et cetera, et cetera. They're all based on this. But this is just one neuron, but you can make a network, okay? On the other hand, this classification learner gave you all the non-neural network options, or the so-called classical classifiers. So what do you see in this video you just played?
Whether you're an expert or
so you define, you train your data at end, you create a function. So if you look at here in the video, so that's how you can use. That's the place I point to. So at the end, you create a trained model. So for this example, the trained model is your fixed, your W values as your predictor. Here is trained model is through this learning process, you decide, okay, we can use this trained model or the parameter already set up to predict function. Then once you give the input data, it will give you a prediction. And this function can be also set up with the interface, whatever. You can translate MATLAB code into C or other languages. You can start to use this as long as you know how to get the data put in here and become vector, and you can make a prediction. Okay, so this and this fixed W here, they're all predictors. They're models, set up models, trained models. So we just found, maybe we'll just go through this classification learner and see how we can use it. We need to prepare some input data, okay, for training purpose, to demonstrate the training. So I'm going to clear my workspace, then come back to this. We are going to use example data which is from the so-called patient data. Anyway, so I don't have to format it. You can you can just copy these 3 lines of code. Okay, so we are going to use information in X, that's your input, then we predict the gender, which is in Y. So I have nothing in my workspace right now, but if I load the patient information, I have an X, the variable. In my X, I have 100 samples, okay, including the age, systolic, this blood pressure, height, weight. So these 4 parameters will help me to decide whether this sample is male or female. I do have the true answer, and that's my, because I know the information input and then know the gender. So this is the training data, okay? So we are going to load X and Y to give to the classification leader, so it can start to train the data, train the model. Okay. So we already prepared the data, so we can load from a workspace. We select input X, okay. Then response. So that's our input, and this use columns as a variable. That's right. So just make sure it's, you know which one is a sample, which one is variable. Then from a workspace, we select Y. Okay. That's your response. That's your target result. You want to predict. Then the rest of them should be okay. All the 5 columns. Sometimes you know you don't want to use one of these columns. You can exclude that particular columns. But in this case, we want to use all the information: height, weight, blood pressure, systolic, all this information. So we keep all this variable name checked, okay? Then you can select here to do the validation. You know what a CV is about, the cross-validation. We can run 5-fold. If you want to run 10-fold, you can change this number to 10. And that's how many times you do this replication in order to repeat the CV cross-validation. I think that's all we need to know. Then you can start the session. Okay, first you have some summary show. So this is column 1 and 2. Depends on your input. Our column 1 and 2 are age and dystolic. So not very informative in any ways. So the age. So if I do 3 and 5, so this 2 parameter look like they correlated. 3 is in our input is height and weight, the 5. So so this scatter plot gave you some quick visualization, the relationship between variables. And the height with systolic. It's positive correlated, but not so strong. But you can do this pairwise scatter plot to look at the relationship between pairwise variables. Okay, so if you have many variables, so in our case we have 100 samples and only 5 variables. We don't need to do any feature selection or PCA to extract top PCs. We can just use them all. But sometimes if you have hundreds variables, numbers larger than your sample, you may want to compress that information using, for example, only the top 50 PC in order to make a prediction. Not using the raw variables, but use some combinations of variables, rotated. So in that case, you can select the PCA. There are other feature selection methods and you can use. For our small example here, only 5 variables, we don't need to do this feature selection. Just keep everything. This is optional, right? So now we are going to move to the selection of the models. So if you click this first button, so if you open this drop down panel here, you can start to recognize this classification method we discussed. We talked about this nearest neighbor, KNN. We haven't talked about this SVM and the kernel. That's for the next week. We do have logistic regression, LDA, discriminant analysis, linear. Okay, so if I decide to use this, I just select. So this is the training plan you are going to use. For example, linear discriminant analysis. I can select KNN. I can select their neural network options. Yeah. Okay, so I select 3 different classification algorithms, LDA, KNN, and the neural network. Once I'm ready, I can just click the train the model. So the 3 methods I selected will run one after another. So the algorithm will tune the parameters just like we guess these combinations of parameters, and validate the trained model using tenfold CV, because we select tenfold, and then compute the overall average accuracy. So you see this is 100%, 96%, 100% by neural network, linear discriminant. And you can start to look at the results. And this is the first linear discriminant analysis with confusion matrix. So there, in our sample, we have 53 females, 47 males, and LDA can classify them correctly 100%. There's no false positive, false negative. There's no number in this confusion matrix in the diagonal position. In the off-diagonal position, there's no mistake, and everything is correct, been predicted after training. So K and N have two mistakes in assign this male into the assign female into male and the two male into female. So there are two mistakes, four in total, and in the KNN algorithm. Neural network did a perfect job as well. Okay, that's a confusion matrix for the ROC curve. Because it's a perfect, there's no information. The model have this very high true positive rate. This 100%, and a very low false positive rate, 0. So you have this curve which is perfect, which in the KNN case, your ROC curve is not as good as the other two models. There's some mistakes. But this ROC curve is only use one setup. It's not like you choose different combinations of parameters. But this is a way you can have a quick understanding of this, how good this model is. And you can also have the result table. Okay, so if you don't know which to choose, they're getting started, then you can choose them all, or you can choose all the linear models. If you click this, all the linear models will be selected, and to run, you can have logistic regression. So select them all. So if you do this, you will have all the models. I select all the models. Let's just keep running and try them all. So in your real applications, you probably want to try them all and find the best one to set up your prediction machine. Okay. Yeah, so the model is still running. We can take a 5 minutes break and let's let it stop. Okay, so this is a very small example, so we don't need to have a long time running to train the model. Sometimes if you have millions of information you want to train a very sophisticated model, you probably want to click this using the parallel options, because it allows you to run this multi-core CPUs, and sometimes you have a GPU support graph computing unit that you can utilize this power to run this training. A lot of this, because for example, this is a 10-fold CV, they can run in parallel instead of one iteration after another. You can run 10 at the same time if you have 10 units. So it's not so obviously we need to use this for small examples. Also, this whole classification learner will be very useful if you have your own data, or you want to make a reusable classifiers and make applications for your future work to do this kind of things. For example, if you tried all the models trained and they found, okay, I want to deploy this particular model. I found its algorithm is good, result is good, so I want to make this reusable. In this case, you can select this model and say generate the function. So you can see, okay, I want to click this, and that's your MATLAB function has been generated. You can save this particular function. So next time you have the trend with the new data. If you want to train, you don't have to come back to this interface. You can just run the function in the command line window. You can also see this model. We can export the model and to save this prediction, the trained model, into the workspace. Okay, so this just saved this linear discriminant model, which has been trained into the workspace. Now, in the workspace, I have this trained model already. Let me my input x. Okay, I already have my x in there. So if I do this trained model, do the prediction function, predict function to give the x. So our x is our training data. So this have 100x samples. It will give me the answer. This will be 100% correct because we trained this model using this information from x. But if you have a new data from a new sample, we can just say, okay, for example, the first one, it trained as a male, it's already in the training model. So you can have a new input, then the model will give you the prediction. Number 15 is female. Any questions for this part? No? Now I will try to see, okay, whether I can, there's, okay. Try to project this laptop into. Okay, let's try. Okay. Yeah, so I can do extension here. Okay, so what I can do is to show you this perception built-in function in this neural network toolbox, because you don't have it, and I don't have that in this machine, so I have to show you in from my laptop here. So that's the function. A small example is like this. So your x have 4 samples, okay, and your target is 0, 1, 1, 1, okay. So this hour. So you don't have to do anything. I just watch, and because you don't have that function in there. So I'm going to give x some So the first point is 0, 0, the second point is 0, 1, third point is 1, 0, and the last point 1, 1. So this classification, the result will be 0, 1, 1, 1. Okay. So to do the perceptron, we will prepare a so-called a net. Okay. So you prepare this net. So perceptron is kind of a very simplified network, neural network. And then we can just train this net by using this input X and output T. Now the net is trained. Okay, so it's doing that. Then these are the parameters. And this is like a diagram of this very simplified network, which contains only a single neuron by integrate information from your input and the weight matrix here. So there's a weight vector here. And then they're constant, basically constant to centralize your data. And then you have a sum function to give you output. This is not a sum function, it's a stem function, and the 0, 1s give you. So that's the perception in the neural network toolbox. Well, if you have a more sophisticated network, you will have a multi-layer of neurons connected to complete the more sophisticated tasks. Okay, so that's this part. Now I'm going to try this example. I think I just play this.
Hi, my name is Joe Hicklin. I'm a senior developer at the MathWorks. I've been experimenting with deep learning with MATLAB and the Neural Net Toolbox. I've written a simple little program that does something pretty cool. I'd like to show that to you now. What I've done is I've taken a webcam and attached it to a neural net that recognizes images. So now I can point the webcam at various objects and it recognizes it. A corkscrew, or maybe a screwdriver, or perhaps a revolver, a harmonica, a teapot, a shovel, that sort of thing. It's not perfect, but it does a pretty good job. So let's look at the code that does this. This is the whole program right here. It's just 11 lines. We're going to go through it, but we're going to do it in 3 separate stages. We're going to clear the workspace. We're going to ask the camera to connect to the webcam, have the camera take a picture, and then finally show that picture on the screen. So when we run this, we should get a fresh picture going on here. And that's the picture we just took. all right? But that's a still image. We want this to be in a continuous video. So we're going to add 3 more lines to put it in a loop. We'll put a while loop around the code that's taking the picture, and we'll add a draw now so MATLAB draws immediately. When I run this, we'll get the same thing, but now it's a live video. All right, finally, we need to add in the neural net. I'm using a network called AlexNet. AlexNet is a large, deep, convolutional neural net. They trained this network on over 1 million images, and it can recognize about 1,000 different objects. I've downloaded it, and now we're ready to use it. This line is going to ask the network to classify the picture we just took. So we'll pass each picture to the network, and it will return a label of that picture. Before we do that, we have to resize the picture to the size that AlexNet expects. It was trained on a particular size of image. Finally, I'm going to use that label in the title of my picture, and I've got to convert it to a string with this command right here. So we're all set. Those are the lines. Let's run it again, and we're running again. I can recognize a keyboard or maybe a space bar, recognize my mouse, or maybe it's a spatula.
Yeah, so when the webcam is pointed to this mouse, you see the title will change because the label has been given to the input image.
And there we go. I hope the simplicity of this program encourages you to try out.
In the network will be given. So where the information, the trained model is inside this called AlexNet. So this is a trained model. Okay, this trained model will not take a vector of the variables, just like our small example to predict a male or female. In that case, we gave 5 numbers, but here the input is a picture. Okay, the picture is also some kind of matrix, or you can vectorize it. But anyway, so you gave this input, and utilizing this trained model to do the classification, and the label will be come back and put in the title of that image. So that's this. See whether I have this webcam. So I copy this and paste into my MATLAB. I may not have So I load a smaller network, okay, because of the limitation of my laptop. So I will see if I can run this to give the labels. Yeah, consider me as a lab code. So it's somehow working, but it's not really good examples. But yeah. There's a yeah. So phone. iPod. Not bad. Yeah, so I believe you need a very clean background rather than this. I try this to the clean background. Clean background, then try tissue round. So you get the idea. So that's using trained model. So you just download that trained model. So if you can make a trained model, then you can share with other people. As long as they know how to use this function, they can build those things. Okay. All right, so we are going to switch back to so what's on our agenda next? Okay, so we are going to learn how to merge to SCE, the dataset we download from using the MATLAB, okay? So I clean my workspace and using GA2 to download two datasets separately. So what I can do is to, for example, the first, I just load one example data and then say the process, and I save this to workspace. So I call this SCE1, so the first data set. Any questions for this part? So I just load example data and save to there. Now I'm going to download another data set. Hopefully it's also from a mouse. And then I will create SCE2. So this is SCE1. So this variable contains all the information about this particular sample. Okay, you have the same problem with multiple gene share the same name. Anyway, so this is the downloaded data, and you can we can process it and using the single click type of annotation. Okay. Let me check whether it's mouse data or not. What you can do is you look at the name of genes, and we should be able to work out this is a mouse gene. This is good. So we know this, the sample is mouse sample, and then click this quick run, and select a mouse t-SNE embedding with annotation. Okay. So with the QC and filter, everything will be done for you automatically. Any questions for this? This can become a routine, because any time you found a data set you want to test, you can just download it with that accession number and quickly see, okay, what cell type in there and what features for these examples. So I have this annotated. I can now click save as the same as what I did for that particular example previously. So I can see this will be SCE2. Okay. Now we prepared two data sets. They all has been temporarily saved into SCE1 and SCE2 in the workspace. We are ready to merge them. They are unrelated so far, and as far as I know. But they share the same gene name, so you can concatenate this with another cell. And is that clear? So what we are doing here? So I really don't need this, so I delete this variable, delete. So I only have two variables, SCE1 and SCE2 in my workspace. Now we select the same import data button, and then I click this import SCE date from the workspace. So now we can select both two samples. If you have 3 or 4, you can also select them. Then you will merge more than 2 samples. So now you click OK, and you will see there are 2 variables, SCE1, SCE2 will be highlighted. And you just click with the shift hold, and then you get both of them. And then you have to decide what you want to intersect or union to merge these two data sets, because some genes may only appear in one sample. SCE1 have one particular gene, which in SCE2 there's no expression, so it has been removed. In most of the case, if these two samples are highly comparable, intersect will be okay. You only take those genes which appeared in both samples, unless you really want one of gene which only showed in one sample, not in another, but you want to keep that gene. In that case, you use union. What that union will do is just add empty genes for that gene in the sample in which this gene is missing. But in most of the case, intersect should be okay. So that's what I'm going to do. So I'm going to intercept this. And now there's only 46 genes left after intersection. Seems this two sets of data is completely different in their gene site. So the only 46 genes overlapped here. So let me see what those genes are. I believe one of these sample is human data, so the gene name are different from each other. But that's so after merge, you have the 5,000 cells, which each is only 2,000 cells. Let me see what the gene name, SCE1G. So that's the gene name from the first sample. 2 from second. It's interesting. There's only, because there's 2 species, so there's no shared gene name. Yeah.
You apply the same system to the same experiment but different groups.
You can, yeah. Mm-hmm. But in that case, you don't have those issues because here, that's mouse gene and not comparable with the human genes, and because of this, when I do this intersection, it's case sensitive. So PSD, the small case, will not match with the PSD with all capital. So I don't have a very good illustration here because this SC1, SC2 from 2 different species. I think I will download another one to see whether, try this example, see whether it's human or mouse data. So now I'm downloading SCE3, the third data, and see whether it's from mouse or human, and then I will decide to merge with which other samples. Click this. Oh, I don't know, so I need to check the name of genes. So all capital, so this is definitely the human sample. So I know I can run this by select human to perform all this. So once it's done, I will save this as SCE3, and I will merge SCE1 and 3. They all from human. Okay, this is my SCE3, has 18,000 genes, and a lot of genes, and I export this SCE data into workspace and label with the number 3 here. Okay, so now I have 1, 2, 3. Don't need this, delete. Now I can come back to my import data and select this load import SCE data from a workspace. I select 1 and 3 by click Control key. By now 1 and 3 will be selected, intersect. The results, okay, the result we have 6,503 genes and 14,000 cells. Now I can click this label to see batch ID, and issued from different names. Okay. So week So week 14 and week 8, they all annotated, so they have labels. But I know this orange one should be from the 3. So you can see, okay, yellow and the blue are from SCE1, orange from SCE2. I can do the embedding again. So T3D. So normally after you merge the data, you need to re-embed it because they have not been embedded correctly. You merge them, you need to run this TSNE or UMAP again to get the idea about the separation. So blue and yellow is from sample 1, okay? And orange is from sample 2, ICE3, okay? So the one we just download have more cells compared to the small example we just use as a demonstration, okay? Any questions to do this? Let's see cell types. They are very different with what we downloaded from a liver, I believe. They are hypothesized fibroblasts. The small example we had is from some immune cells, some endothelium cells, endocrine cells. They are from a beta pancreatic. So that's what you see the difference. It's two very different samples have been merged for demonstration purpose into one sample. Okay. They're not very comparable, so you not compare fibroblast against from one sample against the endothelium cell in another sample. But if they are from a similar tissue, they are comparable. Okay. Yeah, so I believe anytime if you found a good picture, you can say, okay, I want to save into the PowerPoint. This button is supposed to give you the screenshot and to give you into the PowerPoint. This rely on some other toolbox. On this machine, we haven't have that installed. You should also be able to make a video snapshot. This is supposed to give you an MP4 file. So you rotate your sample and take a look, and then you can share with your collaborators. Say, okay, we get a lot of neurons. That's what we need to let them to have ideas. So share this MP4 to your collaborators. So any others? So you know you can change the shape to change the representation or the color schema. Then if you want to have a clear picture, so you can select the view or plot, and then you have a very clean publication quality. So you see, okay, this is TSNE. So you will have this clean picture that you can save as a image or SVG or PDF. for publication purpose. Okay. This is the one I just generated. Okay, so now we may not have a time to set up the Python environment, but I show you, you can give it a try. And so if you do external, then you should have this Python tool, and we need to set up a Python environment. And if you click this, if this has not been set, the program will ask you where is your Python command, executable Python environment, this command line file. So you need to do a search. If you don't have that, so we don't have that there. The same as last time, we try to set up our environment. In our environment, we try to find this particular R executable executable file, so you can search, and it's called R script or R itself. So we set up R, so we have to set up similarly for Python to find the Python executable file. But I haven't installed Python yet, so I don't have this in my computer. So I need to go here, and I don't know which version I can use for this machine. I will search for the software center. See whether the Python has been prepared for me to install. So I search Python. So I do have Python 3.12, 3.16. So I have multiple different versions of Python I can install. So you need to come back to here and to visit a page. There are Python and MATLAB compatibility table. So if you click this link, you will find this table. And we are using MATLAB version 2025B. So this will be comparable with Python version 3.12, okay? Or lower. So we cannot, if we install 3.13, that won't work. So we need to install the one version lower, because when Python publish a new release, MATLAB try to make their product comparable with Python. Well, Python already released 3.13, but MATLAB is still one version older, 2025B. The older version we are using cannot communicate with the new one, so we need to install this slightly older version. So once we have this done, and we should be able to find where our Python executable command is installed. And then we link that command line into the MATLAB, then we set it up. Okay, I think that's all for this week. We'll see you next Tuesday. So, by the way, once I install the Python, I can do the command line cmd here, and I hope I can find the Python. Yep, so this has been already installed, so I need to remember where my Python is installed, and from MATLAB I will point to this place. So from here, I need to set up my Python environment using, oh, it's already here. Yeah, so already here found it. I didn't use this one, but it's selected. It's in the program folder. Okay. Okay, so that's all for this week. Okay.